Thank you, sir, for the very kind introduction. And at the outset, I would like to thank Dr. Dhiman, the director of the Institute, Dr. Ghoshal, Dr. Chandra Chaturvedi, for inviting me to give a keynote address on this very special day, which is the research day of the Institute. Dr. Dhiman started it 3 years back. I still remember, and I was there for the first research day. And I think it's a great, great opportunity for all the students and faculty to showcase their research work. And it's a very nice trend that he has started in the Institute. I am very happy to be invited because this is where I belong. I always think that I am on deputation for a defined period of time to the Ram Manohar Lohia Institute, but this is really where I belong. So it's so nice to come and talk here. As already told by him, this is a very passionate subject of research for me, the potential of stem cell research in medical sciences. So to just build up a little bit on the basics, what are stem cells? And I always say that these are mother cells. This is the way I coined the definition of stem cells that can make any cell or tissue of our body. And the two cardinal properties of stem cells is their ability to renew themselves so that the pool is always maintained and to produce more differentiated or specialized cells of the body. If you look at the potency of stem cells, you have totipotent stem cells, pluripotent, and multipotent. So totipotent stem cells are those where you have the zygote, the fertilized ovum and the first two divisions. The cells that are formed are the totipotent stem cells. And the beauty of these cells is that they can form the placenta. And therefore, they can form the embryo and the whole organism. But what we really deal with are the pluripotent stem cells. They can form, the classic being the embryonic stem cells. They can form any cell of the body, but they cannot form the placenta. And therefore, they cannot form the embryo or the whole organism. And the classical example of pluripotent stem cells is the embryonic stem cells. It's the zygote. After the first few divisions, there is a blastocyst, and the inner cell mass, the cells from this zygote with a few divisions, are the embryonic stem cells. And then, of course, every tissue of the body has multipotent stem cells, which can only differentiate into some limited cell types. For example, you have the neuronal stem cells, the hematopoietic stem cells, the mesenchymal stem cells, etc. So this is what I said, that in the developmental, if you see the development, these are the embryonic stem cells, which are pluripotent. Then you have the adult stem cells, which are multipotent. And in between the two are the fetal stem cells, which can be derived from the fetal tissue or from cord blood or placenta or amniotic fluid, etc. All these are the fetal stem cells. And fetal stem cells are like in between the embryonic stem cells and the adult stem cells. So they are highly multipotent. They're not pluripotent per se, but they are highly multipotent. So why is stem cell research so important in medical science today? One, for tissue regeneration and repair, because of the plasticity of stem cells. Second, you don't really need to do animal studies these days. You can produce hepatic cells, neuronal cells, kidney cells, etc. from stem cells and test drug toxicity on these differentiated cells. So it's very important for testing drugs and chemicals. And very, very important is that they allow us to study the development of an organism. So they've shed a hoard of light on developmental biology. And then they have given us great insights into our own genetic machinery. So this is something I wanted to actually emphasize, what is known as induced pluripotent stem cells. You know, in embryonic stem cells, there's a lot of legalities involved. You get them, they are pluripotent. You get them from supernumerary embryos from IVF clinics. But then you have to take the permission of the parents. There are legalities and ethical issues involved, and they can be rejected by the body because one is giving them across HLA barriers. So there is a new technology for which Dr. Yamanaka and Dr. Gordon were awarded the Nobel Prize in Medicine in 2012. And this is one area of work which was awarded a Nobel Prize within 5 years. They started in 2007 and they were awarded the Nobel Prize in 2012. And the technology here is that you take autologous somatic cells from a patient, for example, the skin fibroblast cells. So they are somatic cells, but you reprogram them. And you reprogram them with these factors like OCT4, SOX2, KLF4, MYC, reprogramming factors. And you reprogram them to become pluripotent. So at the end, you're getting pluripotent stem cells similar to embryonic stem cells, which can differentiate into any cell type in the body, but they are autologous. They're coming from the same individual. So A, there are no ethical issues, no legal issues, they are not rejected. And this technology of iPSCs has actually revolutionized medicine. It has shed so much of light into the developmental biology, into understanding the genetic machinery, and has a huge potential for regenerative and repair therapy also. So as of today, what are the therapeutic applications of stem cells? The only application which is established is hematopoietic stem cells for what is known as hematopoietic stem cell transplantation or bone marrow transplantation. Everything else is under a clinical trial, whether it's for tissue repair, regeneration, diabetes, cerebral palsy, Parkinson's, anything. And therefore, it is very important for people to understand that any stem cell therapy except bone marrow transplantation or hematopoietic stem cell transplantation has to be done within clinical trials with the approval of the institutional ethics committee. You cannot charge patients for it. The way it is being done in India, that every other day in the media, you'll say a patient has been cured of Duchenne muscle dystrophy, etc., by stem cells. And they are charging 8-10 lakhs per therapy. That is unethical. They all have to be done within clinical trials. So how did our journey in the Department of Hematology at SGPGI start in stem cell research and therapy? Way back in 2002, we had a patient of severe aplastic anemia who actually required a bone marrow transplant. First day after transplant, he developed a very rare complication, which is known as Biomyositis. He developed an inflammation and infection of his left thigh muscle. And ultrasound-guided aspiration of the fluid grew E. coli. And soon he developed a Biomyositis of the right triceps muscle also. Obviously, there was bacteremia and the bacteria seeded in the right triceps muscles also. So he was very sick with a 0 blood count. We kept him alive with granulocyte transfusions. That's the time when I standardized, granulocyte transfusions also. Finally, he engrafted. He took up the foreign graft on day 40. and developed his counts back, including the white blood cell count. And when the counts came back, the muscles were teeming with pus, because pus is basically formed because of neutrophils. So at that time, the endocrine surgery department, that was Dr. S.K. Mishra, and I think Dr. Gaurav Agarwal also, we called them into the bone marrow transplant unit, and they aspirated, the pus was aspirated from the thigh and triceps muscle, but the muscle mass was completely necrosed and had to be excised off. But surprisingly, when we discharged him, a large part of the excised muscle mass had regenerated back. And that is what set my mind rolling, that hematopoietic stem cells can regenerate even muscle cells. And that's the time when in world literature, the reports started coming in of the plasticity of stem cells. And that's where we began our journey on stem cell research. And the most common type of stem cells which we have used and world over people are exploring for therapy are mesenchymal stem cells. So these are non-hematopoietic cells with very high ability to divide, and they have the ability to differentiate into cells of mesenchymal origin, mesodermal origin, but also into some cells of a non-mesenchymal lineage. And the most beautiful part of these cells is that they have immunomodulatory properties. They produce an immune silence in the body. So they can be given across HLA barriers, that is from a third party. And you inject them intravenously, they'll go to sites of injury and inflammation because of the secretion of, because they express receptors which make them home to sites of inflammation and injury. So they form a very attractive cell type for cell-based therapy. And the sources, the most common source for obtaining mesenchymal stem cells is bone marrow. Other than that, adipose tissue and the extra embryonic tissues like cord blood, placenta, amniotic fluid, etc. And it was very easy for us to get them because we are routinely doing bone marrows. So taking just an extra aliquot, or when we do bone marrow transplantation and we take the normal marrow, we can get mesenchymal stem cells. But we very soon switched our approach to fetal mesenchymal stem cells. Because if you see, adult mesenchymal stem cells have a variability of their functional characteristics and numbers depending on the age of the donor. And we found a lot of variability in their results. So we said, why not look at fetal mesenchymal stem cells? And when we started working with fetal mesenchymal stem cells, we actually thought that they were wonder cells, because my PhD students used to tell, they were so excited that when you, Dr. Shrikant, when you plate them at night in the flask, by next morning, the whole flask has grown cells. They multiplied so rapidly. And as I highlighted, these fetal mesenchymal stem cells can be got from extra embryonic tissues like the amniotic fluid, the amnion, from the placenta, from the chorionic sac, Wharton's jelly, cord blood, and also from fetal tissue, like fetal cardiac stem cells, fetal kidney, neural, liver, bone marrow, fetal bone marrow stem cells, etc., from aborted fetuses. So our first So the first study that we did was on fetal cardiac mesenchymal stem cells. Means these were aborted fetuses, which we got from the gynae, from Dr. Deepa Kapoor, from the General Hospital. At that time, there was no maternal and reproductive health department. So we got them from the GH, these aborted fetuses with the consent of the parents, and we characterized them. And we actually found that these, from the heart, the stem cells they were expressing all the cardiomyogenic genes. So we then went on to do this study of fetal cardiac MSCs in a rat model of MI, And this was done in collaboration with Central Drug Research Institute, where a rat model of MI was set up, with the ligation of the left anterior descending coronary artery. There was a huge mortality, but then, with some innovations, we could get the mortality down. And in that rat model, we injected the fetal rat mesenchymal stem cells from the heart. And I'm just showing a little bit of the data that this was looking at the cardiac functions, the ejection fraction, the end systolic volume, the end diastolic volume, and the left ventricular myomass. And in the subset of rats, in that arm where fetal cardiac mesenchymal stem cells had been given, there were significantly superior cardiac functions after therapy. And the same thing when we did the histopathological sections, you can see how beautifully you can see the difference. This is the saline group, where you can see there's a lot of on Masson tricon staining. There's a lot of fibrosis. And this is the fetal cardiac MSC group, which after injection you can see is almost a normal looking myocardium. And this is TTC staining, which again stains the infarcted part of the myocardium. And this is, here you can see there's a lot of regression in the infarcted area. So this kind of made us realize that these fetal cardiac stem cells, the fetal cells from the tissue have a very good regenerative potential for that particular tissue. But then, there is a lot of, again, ethical issues and legal issues involved in taking aborted fetuses. So we said it may not really work out from a therapeutic angle. So we started looking at the extra embryonic tissues, and actually now we've switched to amniotic fluid stem cells. And this was just a pilot study which was done. I think Manali is here, she and Dr. Tripathi joined me in doing this study, where we looked at the cardiomyogenic potency of the amniotic fluid stem cells. This was just a pilot study and compared it with that of the bone marrow MSCs. And you can see that the GATA4, the NKX, 2.5 and ISL1, these are cardiomyogenic genes. They were much higher expressed in the amniotic fluid stem cells in comparison to the bone marrow mesenchymal stem cells. And the same was when we induced a differentiation into the 3 subtypes of cells, that is the cardiomyocytes, the endothelial cells. I mean, you can see that the expression of these, they are much better differentiated, the amniotic fluid stem cells, in comparison to the bone marrow stem cells in all the 3 subtypes. So this kind of brought us to the point that amniotic fluid may be a very good source of stem cells. From here, we moved on to looking to amniotic fluid stem cells in a cisplatin induced rat model of acute renal failure. And this we did along with the nephrology department. So we took cisplatin and induced acute renal failure in a rat model, injected amniotic fluid stem cells. And here is the healthy, you can see that the creatinine level gradually in the AFSC treated group comes down by day 12, significantly comes down. Initially, it is comparable between the saline treated and the amniotic fluid stem cell treated group, but then it comes down. And if you look at the histopathological analysis, this is how the control group and the cisplatin injected group looks like. And this is the comparison between the saline and the amniotic fluid stem cell group looks like. And you can see that there's a huge difference in the reversal of the histopathological changes after giving amniotic fluid stem cells. So this again brought us to the point that these amniotic fluid stem cells are kind of not by definition pluripotent, but they have a very great multipotent property. And this was in diabetic rat model. This is unpublished data. We are yet to publish it. This was done along with Dr. Leena Rastogi. And here this is the diabetic mouse model, streptozotocin induced. And this is how the blue one shows that this is a normal rat. This is the way the blood glucose levels, the urine volume, the 24-hour water intake. These are some of the parameters to judge the diabetic status in these rat models. And this is the body weight. So the blue lines are the normal rats, and then the green ones are where the stem cell therapy has been given. And the red is the diabetic rat where no stem cell therapy has been given. So if you compare with the red, there is a significant improvement in that arm of the rat model where stem cells were given. So I just in the end would like to highlight this one case again of bone marrow transplantation, who after COVID infection presented with persistent fever, bleeding gums, passage of black colored stools. He was diagnosed to have a severe aplastic anemia. He had to almost undergo an emergency hematopoietic stem cell transplantation in view of the severity of the aplastic anemia that he had, and he had a HLA match system. The transplant went very nicely till day plus 12. The patient engrafted, but on day plus 23, he developed a severe acute GI, gastrointestinal graft versus host disease, with 18 to 20 episodes of loose stools per day. This is a known complication after bone marrow transplantation, where the immune cells of the graft mount an immune reaction in the recipient cells, in the host's body. And it commonly affects the liver and the gastrointestinal tract and the skin. So the patient had a profuse diarrhea. So he was administered steroids, but we found no response. So he was labeled as steroid refractory graft versus host disease, which almost always has 100% mortality. So we kind of debated what we should do. And we had nothing to lose, so we gave mesenchymal stem cells, third party, human mesenchymal stem cells derived from the cord blood. And the diarrhea started regressing within 24 hours. You could see that the volume and the episodes came back, and in about a week's time, his graft versus host disease had completely regressed. So this case also highlights the potent immunomodulatory effects of mesenchymal stem cells. So this is how we have proceeded with mesenchymal stem cells in humans, and of course the trials in amniotic fluid cells. And now Manali, who is sitting here, is looking at the c-KIT positive amniotic fluid stem cells for cardiac regeneration. So I feel it's a very, very important source of stem cells, and Dr. Neeta has been giving us the amniotic fluid. In the last, I just wanted to share this very beautiful paper which was published in Nature 2022, where a synthetic embryo with brain and beating heart has been developed by scientists in UK from multiple stem cells. And these are researchers from the University of Cambridge, where they have created a model embryo from mouse stem cells that can form a brain. Till now, they've shown a brain and a beating heart, which is the foundation of all other organs of the body, a new avenue for recreating the first stages of life. So this is the way that stem cell research is globally going. I found it a very exciting and a very powerful paper, so I thought I'll touch on it in the end. So what is the future? No doubt, stem cell research is at the cutting edge of medical research and has promises for the treatment of several diseases for which no treatment exists to date. And it also is creating organs for transplantation. But we have miles to go and should not lose hope and continue our sustained efforts in investigating the therapeutic potential of stem cells. Thank you, and I want to acknowledge the Department of Biotechnology and my entire stem cell research group for all this work that we have been able to do. Thank you very much.